In this chapter, we're going to focus on cytology, the study of cell, to discuss the function and the structure of cells. Your body is a cooperative society of trillions of cells. For human body, it is composed of about 36 or 37 trillions of cells. That's a lot of number of cells. If you stacked up 8,000 cell membranes, they would only be as thick as a page in this book. That means for your textbook. Every second, you produce about 2 million red blood cells. A single nerve cell in the human body can reach up to a meter in length. That's the longest cells in the Earth's ecosystem. And the cells of a whale are about the same size as the cells of a mouse. All the living organisms are made up of cells. That's the cell theory. All the living things, they are composed of cell. Cell is the basic unit of living activity, that's why. The cell is the simplest collection of matter that can be alive. All cells are related by their descent from earlier cell because they are born by the cell division from their mother cell. That's why. Cells can differ substantially from one another but share common features by their DNA. And the cells are too small to see with our naked eyes. We cannot see. So that's why we need microscope. So the development of cytology is followed by the development of microscopy. And microscopes are used to visualize the cell. And very commonly used microscope we call that's the light microscope, LM. Visible light is passed through a specimen and then through the glass lenses. Lenses refract the light so that the image is magnified. And when we talk about the qualities of microscope, there are three important parameters of microscopy. First one is magnification. How big the image can be enlarged. Magnification, the ratio of an object image size to its real size. So it is usually expressed by times 10, times 100, times 200, something like that. Only the magnification, that's not enough. We have to see the object, the image, clearly. We call that the resolution. The measurement of a clarity of the image or the minimum distance of two distinguishable points. And the third one, that's the contrast. That's the visible differences in brightness between parts of the sample. And we can see some examples of size or scale of cytology. So usually we can use the unit of length as a micrometer. Micrometer, we can see 100 micrometer. What's the micrometer? So 1000 micrometer make up 1 millimeter. So 1000 micrometer is equal to 1 millimeter. So we can see a 10 micrometer for the nucleus, and the most bacteria and mitochondrion is between 1 micrometer to 10 micrometer. How about the below the micrometer? We can see what? Nanometer, nm, yeah? Nanometer. So when we have 1000 nanometer, it is equal to 1 micrometer. Again, yeah? 1000 nanometer is same as 1 micrometer. So we can see 100 nanometer, that's the scale for smallest bacteria or viruses, and 10 nanometer, that's the ribosome for protein, and 1 nanometer, that's the small molecules. Diameter is about 1 nanometer. So nowadays, not only for the biology, but in many fields, they talk about nano. Yeah? Nanotechnology is something. I want you to understand the scale of nano. You have to check up. Light microscopes can magnify effectively to about 1000 times the size of the actual specimen. Various techniques enhance contrast and enable cell components to be stained or labeled. The resolution of standard light microscopy is too low to study organelle, the membrane-enclosed structure in eukaryotic cell. However, in 1950s, the scientists, they developed the electron microscope to enhance the magnification and the clarity, resolution significantly. So the development of electron microscopy extends the range of cytology. So we can observe the structure of the organelle, small organelle, and membrane, and small structure inside the cell. This one showing the image of the light microscopy. So usually the image is kind of thin and flat because basically we make a thin section and put on the slide. under the light microscopy. That's why image is kind of flat, yeah. And we can see some cell, and the magnification and resolution is not high enough to observe inside the cell. So we can see the overall structure of the cell by the light microscopes. And for the electron microscopy, basically we have two types of electron microscope, EMs. The basic one, sometimes we can see the structure, big cylindrical structure of the electron microscope. That's the what? That's the transmission electron microscope, TEM. That focus a beam of electron through a specimen. So it's same thing. Yeah? For the TEM, transmission electron microscope, we make a thin section of sample, and the light, it has go through, transmitted. Yeah? So that's why we call that the transmission electron microscope. So the image from TEM, it is kind of flat because basically the sample, we make a sample and make a thin section for the preparation for TEM. However, very different image we can see from the SEM. It is so-called scanning electron microscope. Focus a beam of electron onto the surface of a specimen, providing the image that looks three-dimensional. So when we look at a three-dimensional shape, because they don't make a section, yeah? So that sample is gonna get frozen just immediately, and we can see their three-dimensional shape by scanning electron microscope. So the basic thing, the first one, that's the TEM, the other one was developed as a scanning electron microscope, SEM, and TEM are used mainly to study internal structure of the cell. However, we can see internal structure inside the cell by TEM. However, their image is kind of flat. Yeah, that's the difference. The three-dimensional structure, the image we can find by SEM, scanning electron microscopy. On the other hand, the image from TEM, transmission electron microscope, is kind of flat because that's the image from a thin section. Yeah? Very, very thin section. That's why the image is kind of flat, two-dimensional. We already defined the eukaryotic cell in previous chapter. So eukaryotic cell, it is characterized by having their well-developed nucleus. So they have real type of nucleus, true type of nucleus. That's the eukaryotic cell. Not only for that, in addition to that, eukaryotic cell, it has many membrane-enclosed organelle. So membrane-surrounded organelle, they have many inside the cell. So eukaryotic cells have internal membranes that compartmentalize their function. They can separate their function by those organelle, by the membrane. So the basic structure and functional unit of every organism is one of two types of cell, as we already discussed over in previous chapter, prokaryotic or eukaryotic. Only organisms of domain bacteria and archaea consist of prokaryotic cell, as we already discussed over, and protists, fungi, animals, and plants all consist of eukaryotic cells. Comparing prokaryotic and eukaryotic cells. So basic features of all cells, they are all common for prokaryotic. In eukaryotic, doesn't matter. First one, the plasma membrane. Prokaryotic cell and eukaryotic cells, same thing. They are surrounded by plasma membrane, cell membrane, and the semi-fluid substance called cytosol. That's the liquid part inside the cell. They have cytosol. For prokaryotic and eukaryotic, that's the same. And they have chromosome carrying genes. Chromosome consists of DNA, that's the genetic material, inside the prokaryotic and inside the eukaryotic. And the ribosome. Ribosome, that's the major structure to produce protein. So that's why prokaryotic and eukaryotic doesn't matter. They have to produce protein for their living. That's why they have ribosome. And the prokaryotic cells, the easier understanding, the bacterial cell or prokaryotic cells are characterized by having no nucleus. They have DNA, they have genetic material, but it is not surrounded by nucleus. So that's why no nucleus. But it does have DNA. So DNA in an unbound region called nucleoid. So the term nucleoid is kind of funny. So in biology, something OID means I cannot call that the something, but it is something like structure. Function is about something. Then I cannot call that something, but function is same as something. Kind of funny, yeah? So then, in that case, I put that OID. So I cannot call nucleus, but the function is like a nucleus. So what can I say? Nucleoid. Or I put OID. That's why. Because by the definition, prokaryotic cell, it cannot have nucleus. But the location for DNA is present. The function is like a nucleus. That's why I cannot say nucleus. But function is something like nucleus. That's why the name is called as nucleoid. Funny. And there are some other biological terms ended up with OID, like embryoid, like proteinoid, something like that. Okay, anyway, no membrane-bound organelle. Prokaryotic, they don't have membrane-bound nucleus. Not only for that, they don't have any other membrane-surrounded organelle. They don't have. And cytoplasm bound by the plasma membrane. So cytoplasm, all the contents inside the cell it is surrounded by plasma membrane, that's the same as in eukaryotic cell. This one showing you a typical prokaryotic cell. That's the rod-shaped bacterium. Okay, first, all the cell, not only for the prokaryotic but eukaryotic same. Yeah? The prokaryotic and eukaryotic cell, they are surrounded by basic cell membrane we call plasma membrane. At the center we can find plasma membrane, so it is surrounded by plasma membrane. And we can find ribosome, some small dot-shaped ribosome to produce protein. And the location where DNA is located, the blue thing, the DNA is located, the location we call that the nucleoid, as we just discussed over. And usually the bacterial cell outside the plasma membrane, they have some protective structure, cell wall, so they are surrounded by cell wall for the protection. And one more time, most of the bacteria, they have another protective layer. Now this time, not very hard material like the cell wall, but it is kind of soft, sticky material. We call that the capsule. So outside the cell wall, they have one more layer of protection called capsule. So capsule, it has a soft protection, and the other thing, it makes it easier to stick to host cell by the capsule. And many of bacteria, they have tail-like structure for their movement. By the moving of those tail-like structure, they move around. So the tail-like structure we call flagellum. One flagellum, 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 and many flagella. So most of the bacteria, they have those flagellum or flagella. And outside the capsule, many bacteria, they have hair-like structure, like we call fimbria or fimbriae. Those kind of hair-like structure, so make it easier to stick to host cell for their living. So for the right hand side, we have an image by the TEM for the bacteria. On the other hand, eukaryotic cell, they are characterized by having DNA, that's the same, yeah? But inside the nucleus, yeah? So DNA is inside a well-developed structure of the nucleus, yeah? Surrounded by membrane. So it is bounded by a membranous nuclear envelope. So it is surrounded by membrane nuclear envelope. And not only for that, they have many membrane surrounded organelles they have. And cytoplasm, in the region between plasma membrane and nucleus, we already discussed over cytosol. That's the kind of or is a liquid part inside the cell we call cytosol. How about cytoplasm? Cytoplasm, it includes cytosol too. So cytoplasm means that's the whole space between nucleus and plasma membrane. All those things between plasma membrane and nucleus, including cytosol, including ribosome, including Golgi apparatus, including what is that? So endoplasmic reticulum, and what else? Mitochondria and chloroplast. All those organelles included, we call that's the cytoplasm. And eukaryotic cells are generally much larger than prokaryotic cell. How much bigger? By the volume, about 1,000 times bigger than prokaryotic cell. That's a lot bigger. The plasma membrane is a selective barrier that allows sufficient passage of oxygen, nutrients, and waste to service the volume of every cell. That's why plasma membrane is very important. It allows to pass through that. Many material has to go pass through that membrane for the transport. As I mentioned, that plasma membrane's area, it is very important because through the plasma membrane, they have to go through transportation from inside to outside, or from outside to inside, like that. So that's why, proper the plasma membranes, membrane surface area, it is very important. That's why cell's volume cannot be just bigger and bigger, cannot be, because they have to have some proper surface area to volume they have to keep. So metabolic requirements set upper limit on the size of cells, so they cannot just bigger and bigger, limitless. It cannot be. The surface area to volume ratio of a cell is critical, so they have to keep certain ratio for the surface area to volume. As cell increase in size, its volume grows proportionately more than its surface area. So as I mentioned earlier, if a diameter of a eukaryotic cell is about 10 times bigger than prokaryotic cell, then surface area for the eukaryotic cell, it can bigger, about 100 times bigger for the surface area of the eukaryotic cell than prokaryotic cell. Then what? Then its volume for the eukaryotic cell can become 1,000 times bigger than prokaryotic cell. Then what? Then surface area to volume, it is what? It is 100 to 1,000. So it can go down, it can reduce down sharply. So they can support, those 100 surface area can support the volume of 1,000, maybe not possible. However, for the eukaryotic cell, they have many internal surface area. By the many surface area of membrane surrounded by many organelles like the mitochondria, like chloroplast, like endoplasmic reticulum, like lysosome, like Golgi apparatus, etc., etc. There are so many internal organelles surrounded by internal membrane. Those internal membrane, they compensate for the reduced surface area of the eukaryotic cell. That's why eukaryotic cell, no problem, yeah, it's okay. For the less surface area is okay, because those surface area can be helped by many internal membrane area. Okay, so next picture maybe can explain some more. This one showing the diagram to increase up cell volume. So for the small cube, one side the length is just 1. When it increases up 5 times, 5, then what? The length from 6 and the surface area increase up into 150, and 150, that's a surface area. How about the volume? Volume gonna get increased up from 1, from 1, the volume, because 1 by 1 by 1, then that's the 1. That is small cube's volume is 1, but 5 by 5 by 5, 125, that's its volume. So the surface area to volume is, for the small cube is 6, but for the surface to volume it is 1.2. It's very low, it's very hard for those cells for their living. However, the last picture, so it has a same volume, but it can increase up many surface area. So surface area, when you calculate 750, so surface area to volume, it is increased up same as small cube, 6. So it's no problem for their living, because they secure so many internal membrane area. So that's the explanation. For the eukaryotic cell, it can increase up by the volume about 1,000 times bigger than prokaryotic cell. A eukaryotic cell has internal membranes that partition the cell into organelles. So a eukaryotic cell has many organelles surrounded by membrane. That's why those membrane has effect to separate the space inside the cell. The basic fabric of biological membranes is a double layer of phospholipid bilayers, so-called, and other lipids. Plant and animal cells have most of the same organelle. Okay, we can see the structure of eukaryotic animal cell. Okay, so eukaryotic cell, so it has to be, the cell has to be surrounded by plasma membrane. So what is plasma membrane? So at the right hand side, you can see plasma membrane. So cells surrounded up by plasma membrane. And inside, we can see the most conspicuous structure. We can see nucleus surrounded by nuclear envelope. And inside the nucleus, we can see some dark area. We call that the nucleolus. Sometimes we can see more than one, then we call that's the nucleoli. Yeah. So instead of US, we put just I, nucleoli. And inside the nucleus, they have to have what? The DNA in forms of chromatin. Chromatin, it is composed of DNA and some protein composed together to make up chromatin. And those nucleus is surrounded by double membrane nuclear envelope. And outside the nucleus, we can see blue colored. So that's not actually not blue colored, but it is expressed as in blue color. But we can see many folded up membrane structure. We call that the endoplasmic reticulum. Yeah. So they put on top portion, yeah, endoplasmic reticulum, ER. ER, it is divided into rough ER and smooth ER. Rough ER means outside the endoplasmic reticulum membrane, outer surface, they have many ribosome attached, ribosome bound, then we call that the rough ER, because when we put under TEM, transmission electron microscope, those membrane, they imaged out as rough line because of the presence of ribosome. That's why we call rough ER. Without ribosome, we call that ER as smooth ER because that line of membrane image is kind of smooth line. So smooth ER. And we can see what we can see inside the cell mitochondrion. Yeah. Mitochondrion have to go through cellular respiration, and we can see lysosome, and we can see many flattened pocket, a series of flattened pocket shape of membrane, Golgi apparatus. And what we can see peroxisome and some surface of the cell. It is making protuberance here, so make something like hair-like structure, something many microvilli. And some animal cell, in their southern stage, they can have flagellum, tail-like structure. And just below the flagellum, we can see centrosome. Centrosome, it is composed of small two cylindrical structure. They crossing each other at 90 degree at right angle all the time. We call centrosome involving in cell division. And the empty space inside the eukaryotic cell, it is not empty, but it is filled with many small protein fiber. All those small protein fiber, they have some type. The smallest type of those fiber we call microfilament, and medium size, intermediate filament, and the largest size we call this a microtubule. All those things, those protein fiber together make a kind of extensive network of fiber, like a network-like structure. Collectively, we call this the cytoskeleton. Cytoskeleton. So all the empty space inside the eukaryotic cell, it is filled in with a network of cytoskeleton. That's the basic structure for the eukaryotic animal cell. The eukaryotic plant cell, cytologically, plant cell and animal cell are not very different. So many things they are same. So same thing. Plant cell, basically the cell, it is surrounded by plasma membrane. Plasma membrane, that's the same. And they have exactly same nucleus, surrounded by double membrane nuclear envelope. And inside the nucleus, we can find nucleolus, and DNA stay as in forms of chromatin, composed of DNA and protein. And we can find ER, smooth ER and rough ER, and we can find Golgi apparatus, and we can find exactly the same mitochondrion, and empty place is filled with extensive network of protein fiber, cytoskeleton, that's about same. But then what's the difference? Difference is this is plant cell, that's why they have to go through photosynthesis here by the organelle of chloroplast, green chloroplast. Chloro means green, that's why chloroplast. Green body, chloroplast. And this one plant cell, so inside the plant cell, some space is enclosed by the membrane containing mostly water. We call central vacuole. Yeah? Central vacuole. That's the typical for the plant cell. And plant cell, they need protection outside the plasma membrane by strong cell wall. I mean, cellulose, major ingredient with cellulose, they made up a cell wall for the protection. Cell wall. And there are some passage from cytoplasm to next cytoplasm of plant cell, plasmodesmata. Yeah. So when they have just one, we call plasmodesma. And many, more than one, plasmodesmata, like that. So the cell wall and chloroplast and central vacuole, that's the different feature from animal cell.